my cutest little children. I hope all of you are doing great, and I hope am I visible? Let me check. Yes. So I'm perfectly visible, and I hope I am audible as well. Yes, I am audible as well, and cool. So I'm just waiting for all of you to come back. I hope you had a great day at school, and if at all you're someone who didn't have a good day at school, then it's time to change your mood, right? And to make you really happy, I think this particular class is going to do that. By the way, for those of you who have no idea who this person is, I am Jasmine Joy, and I teach biology. And my dear children, today is the third lecture of this particular chapter, that is human reproduction. We have done 2 videos on this. Before this, I have actually completed sexual reproduction in flowering plants, and I have completed them in detail. So each of the parts, I wish you go and check it out once. And in those parts, for example, if I am teaching the structure of the male reproductive system in the case of plants, then in that particular case, I will be also explaining certain questions that have come in KCET. So that is how I go, and you will be seeing that over here in this particular video as well. And as I said, I have done 2 videos on this chapter, that is human reproduction. The first one is the male reproductive system, and the second video which was there, it was the female reproductive system. Today we have the third lecture, and the third lecture is going to be about spermatogenesis and oogenesis. Now this is a topic which makes you go like this, but I'll make sure that you like, oh, such a nice topic, right? Now, Kishore, okay, hello, good evening dear, good evening Jasmine Mam. Can you please tell your old student, where is Ankit sir? He's not coming since long. Please not be worried, you are going to see him really soon, okay? So most probably by tomorrow you will be seeing his face. Cool? Will that work? Okay, now going ahead with the chapter, and I hope I can go ahead smoothly. This is very important. Please know, spermatogenesis is a very important topic, and basically I am going to start it with a story. We are going to study in the form of a story. Okay, so let's go ahead. Spermatogenesis means what? Genesis, if at all we go ahead with the terms Genesis, it means formation. It means formation of something new. And when we say spermatogenesis, it's basically talking about formation of sperms. The sperms have not yet been formed, they're going to form. And there are a few steps that you need to remember, you need to know, and we are going to learn about it. So the first guy that we have in the picture is spermatogonia. Is what? Spermatogonia. I'll say it's the pardada, or basically what? Great grandfather of sperm. Great grandfather of sperm. That is what? You have spermatogonia right over here. Right? Now this spermatogonia which is there, its dream is to form sperms later on. So which is why it says that I'm just 1 in number. First of all, the first thing that I have to do is I want to increase my number, which is exactly why what it does, it goes on dividing itself. If there was 1 cell, now it has divided and have become 2. And this process of division is known as what? It's known as mitosis. You might have heard about mitosis and meiosis in grade 11th or before as well. Mitosis is a division of a cell where it just divides without reducing the number of chromosomes. For example, if I say that 46 number of chromosomes were here in this cell, okay, and this cell divided into 2, basically the other cells will also have 46. It will not be like, Macha, I had 46, you take 23, you take 23. If it goes like that, I will call it reductional division. But over here that is not happening, no reductional division is happening, everything is equal. So I'll say there is mitosis happening. I also call mitosis as what? Equational division. Equational division as in whatever number of chromosomes over here, those many number of chromosomes will be over here also. So spermatogonia basically just divided to increase the number. And over here AK Kishore says, bio is not difficult with you, ma'am, you make it cakewalk for everyone. Thank you so much, and that's so sweet of you. Thank you so much, it means a lot, and that's a senior saying that, and really it means a lot to me. Thank you so much, dear. Thank you for making my day. Okay, now coming back to this topic. So I said spermatogonia had divided. Now, what was its dream? Its dream is to make sperm. But how? So listen to me. When a guy hits puberty, that's when the spermatogonia starts to divide like this. Cool. And when it divides, there was only one spermatogonia, right? When I say there is only one, there's only one. Now, if I say that this guy is going to make sperm, it will be making few sperm. But what about more sperms? We want more and more sperms. So in that case, we should be doing something, right? So what happens is when this division happens, this one guy says that I'll be just like my dad. I'll be just like my dad. I'll keep on dividing. It's okay. You who are there, this is a sibling that we have, and the sibling says that, okay, then I will take up the job of making this sperm. This particular cell which is there is going to undergo some changes out there. It will be undergoing some changes and it becomes the primary spermatocyte. It becomes what? The primary spermatocyte. Basically, this is the one that is actually going to form the sperms. Cool. So over here, this is the primary spermatocyte, and if at all you are asking me that is this cell dividing to form this, I'll say no, it's not dividing to form anything. It's just that there are certain changes that are going to happen in this cell. We say it's maturing. It's maturing in a way, okay, in a good way, and we call this process as differentiation. What do I call it? I call this as differentiation, and I say that this cell has differentiated to become what? The primary spermatocyte. What is the number of chromosomes over here? It remains the same since there was no division. It remains the same. It is 46 only. I hope you have got the logic. If you have not understood this part, you will not understand anything. Okay, so we have spermatogonia where inside the seminiferous tubule where the sperm formation is going to happen, right? The spermatogonia says that, Macha, I am going to divide myself and I am going to increase my number. I actually want to form sperms later on, so it divides. What division? Mitosis. The number of chromosomes of 46 over here, it becomes 46 here also. Okay, now both of them cannot say that I will become sperm, because you need someone to maintain the legacy of being a spermatogonia as well, which is why one cell says that I'll be just like my father. I am the person who probably will give rise to more of these cells. Okay, cool. The other says that I am going to differentiate into something else which is actually going to form sperm, and that is what? The primary spermatocyte. Cool. Now, in layman's term, if at all we talk about biological terms, we cannot say 46 chromosome, 46 chromosome, 46 chromosome. Rather, we have a term for it. Whenever we say the number of chromosomes of 46, we say it's diploid in nature. In the case of human beings, when we say 46 number of chromosomes are there, we say the cell is diploid. That means it has double the number of chromosome. 2n is what? Double the number of chromosome. And when we say that the cell has finally divided and it formed the sperms at the end, my dear children, will not be having double the number of chromosome. It will be having half the number of chromosome. So half of 2n is equals to what? It is just n. It becomes haploid later on. It becomes haploid later on. So here, if diploid is 46, then haploid content becomes half of it. What is half of 46? It becomes 23. So what happens at the end? We get sperms which are having half the number of chromosome, that is 23 number of chromosome. Over here, I have not divided like that. This is just mitosis where the number of chromosomes remains the same. If I started with diploid, it is still diploid over here also. What about this cell? This is also diploid. This is also diploid. When does anything become haploid? When it undergoes another type of division which is known as meiosis. What do you call it? Meiosis. So this was mitosis. Now the primary spermatocyte is going to undergo something that is known as meiosis, and we are going to talk about that right now. I hope this part is clear, and let's go ahead with the next part. As I said, we are talking now about the primary spermatocyte, which is the one which is going to form the sperm later on. It is diploid in nature. That means I can say in human beings it is having 46 number of chromosomes. And I said that it is going to undergo meiosis. You all should know meiosis happens in two stages. It's not like mitosis. Cell divided, it became two. No, it does not. It does not happen like that. In meiosis, there is first division that happens. We call it meiosis 1, where you get two cells at the end. Okay, you get two cells at the end. Now guess the number of chromosomes over here. Will it remain 2n or will it become half? Anyone who is watching the video, you can tell me. I'm not going to judge you. Listen, Jasmine Mam will never judge you guys. And I don't think a good teacher can ever will. No one in this channel is ever going to judge you. Please let me know the answer for this. Do you think the number of chromosomes are going to remain the same? 2n remains the 2n, or probably 2n becomes half of it? What do you think? Any idea? Any idea? You can probably talk about it later on also. If at all you do not know right now, you can check also. Do not mind. Okay, so basically, since you guys are not saying, I'll tell you the answer. Over here, the 2n is going to become half for the first time. Since I said it is undergoing what? Meiosis. And in the first round of meiosis, what happens? The number of chromosomes, they become half, Baba, for the first time. And they are like, oh my God, what have happened? And what do you call these type of cells as? Which have half the number of chromosome. And it's the end product of this. You call them as, this was primary, so now you just name it back into secondary spermatocyte. You call it secondary spermatocyte, and always remember, this guy is the first one to have half the number of chromosomes. Or basically, I'll say it was diploid until this point, but once meiosis has started, it has become haploid. Now, Baba, is meiosis completed right over here? No, ma'am, you only told meiosis happens in 2 stages. Right, exactly right. So what happens? This cell again divides into 2. Okay, this cell also divides into 2. This is the second round of meiosis. Is this a final division? Yes, that is a final division. At the end, you get 4 cells. At the end, you get 4 cells. What is the number of chromosomes going to be like? It becomes half again? Do you think so? No. Only once this half happens, okay, it was 46 here, it became 23. And that's all. We do not want to make it more half. We just want 23. So what happens here? All of them remains the same. They are all haploid. They are all haploid only. What do you call them, Baba? You call them primary, you call them secondary. Now, would you call them tertiary spermatocyte? No, you actually have a name for it. And what is it? Spermatids. What do you call it? Spermatid. And I remember it in a fun way, okay? I actually call it, since it is spermatid, I remember it as kid, kid. So why do I remember it like that? There is a reason for it, because I get questions from it. So what happens is, you might have seen the picture of sperm. Sperm is a very long structure. Okay, it is going to be an elongated structure that is there. This spermatid which is there, it is not elongated. It is global. It is round in structure. Now, do you think this round sperm can move around very easily? No. Why do you think sperm is elongated? Such that the passage can be really easy. It can move fast. That is the reason. But here this guy is round in structure. So the first thing is that the kid is not mature. We want to make the kid to be converting into a mature person out there. So that is exactly what I'm saying. Over here, the spermatids which are there, they convert into something else which looks like sperm. What do you call them? Spermatozoa. You call it spermatozoa. And this process has a name. This process has a name where you probably will be like, oh my God, I cannot remember any more names. It's very easy. It's known as spermio. Okay, it is known as spermatogenesis. Genesis means formation. Spermatogenesis means, Baba, the spermatozoa is being formed. The whole process until now, what we are discussing is what? Spermatogenesis. That means formation of the sperm. Here, this specific process where a kid is converting into a mature guy, it is known as spermatogenesis. Do not forget it. And what usually happens, where do you mess around is that they'll tell you formation of spermatid from spermatozoa is known as spermiogenesis. That will be very wrong. No, you're telling ulta. It is spermatid that converts into spermatozoa, not spermatozoa into spermatid. You get it? So always remember, that's why I said it was a kid before and then it matured. And this process is known as what? Spermiogenesis. Now after spermiogenesis, what happens? The sperm obviously over here is being formed. Where all of it is happening within the seminiferous tubules, right? It is happening within the seminiferous tubules, all of it is happening. In the seminiferous tubules, I'll say sperms are being formed. Along with that, you have cells nourishing the sperms. What do you call them? Sertoli cells. You call them Sertoli cells. So usually what happens, okay, the spermatozoa which are being formed, they are like, oh my God, I cannot take anything up. So they go and embed their head. Okay, they go and embed their head into the Sertoli cells. Like, Sertoli cells, you are the one who promised to nourish me, right? Nourish me right now. Nourish me right now. That is a process that happens. But there will be a time where this papa spermatozoa has to move out. It cannot be here for long, right? It has to move out, right? And that process is known as, it is actually said in your NCERT, the process of, the movement of sperm from the seminiferous tubules is known as spermiation. So aviation, you can remember, it's spermiation. From seminiferous tubules, actually they are going to go to different places. What do you mean by different places? As in rete testis, vasa efferentia, the duct which are there through which the sperms are going to be carried. That is what you mean. Okay, so in general, if you talk about the technical terms, it's not given in any NCERT. Actually, it's just that the sperm's head were there in the Sertoli cells. Now they are just going to move away from the head and they're going to come in the space which is there in the seminiferous tubule. That process is known as spermiation. But according to your NCERT, it says that spermiation is the process of movement of sperm from the seminiferous tubules. That means they are totally out. So you can remember like that. Okay, please remember like that. What is spermiation? Movement of sperm from the seminiferous tubule to outside. Okay, so that will be the last step that is there, that is spermiation. Always, sometimes you might get confused with spermiogenesis, spermiation. So as I said, spermiation, you can remember like aviation. So it is moving out, the sperms are moving out. You just remember like that, moving out from where to where? From the seminiferous tubules to probably the next duct which is there, which is going to carry the sperm. Cool? Okay, it's very easy. It's not at all complicated at times. Okay, at times we feel that, oh my God, what am I studying? It's difficult and all. But trust me on this. If at all you just revise it once, everything is going to be sorted. So I'm just going to do a quick revision and I need all of your attention. Okay. We are talking about spermatogenesis. Forget about all this thing which is there in the background. We are talking about spermatogenesis. That means the formation of sperm. Where does it happen? In the seminiferous tubule. In the seminiferous tubule, what is there apart from spermatogonia? There is one more thing, and that is known as Sertoli cells. Sertoli cells are the one that nourishes the the future sperms which are going to be there. Okay, ma'am, you said spermatogonia, right? What is that? It is the male germ cell, basically the one that is going to divide and form the sperm later on. So suppose there is spermatogonia right over here. This spermatogonia, which is a diploid cell, a normal cell, which is having 46 number of chromosomes, is going to divide. You have two cells now. Okay, you have two cells now. What do you call them? You do not have any name for them. Basically, they also have 46 number of chromosomes. What type of division did it undergo? It underwent mitosis, normal division. Okay. Now, one cell said that I'll be like my father and basically I'll keep on dividing. The other cell said that my father wanted to make me a sperm. I will go ahead and be a sperm. So this cell which is there, it undergoes a process of differentiation or maturation. Okay, you call this differentiation actually. It is not division, it's just differentiated, it gets differentiated. And we'll now call this as what? Primary spermatocyte. Okay, it has 46 number of chromosome. This primary spermatocyte has decided to form the sperm, which means that it has to undergo the process of meiosis. Meiosis happens in two stages, meiosis 1 and meiosis 2. The cell which is there, it divides into two, right? And when I said meiosis, you need to understand it's a reductional division. The number of chromosomes over here gets reduced to half over here. It becomes N and become N over here. For the first time, it becomes haploid. What do you call them now? Secondary spermatocyte. This secondary spermatocyte again undergoes meiosis because, as I said, meiosis happens in two stages. Second stage, will the number of chromosomes get reduced again? No, it remains the same. What do you call them now? They are like kids, mama. They are globular in shape. They are round in shape. You will call them spermatid. Now, spermatid wants to grow into actual sperm. I didn't want to form this. I want to form a sperm out there. Then make it. This process of conversion of spermatid to actual spermatozoa is known as what? Spermiogenesis. Okay, I want to become me. What are you doing over here? So, spermiogenesis, you can remember like that. Okay, finally, these sperms are being formed. These sperms go and embed on the head of the Sertoli cells and say that nourish me, nourish me. Then Sertoli cells one day says that go, you should not be here, go explore the world. So basically it goes on, moves out from the seminiferous tubule, and according to your NCERT, it is said as what? Spermiation. So I remember it as aviation. Until this point, is everything clear to you? I don't think I can go in more depth than this, okay? It's the easiest way to explain, but if there is any person out there who is watching this video and probably not understanding the concept, I can explain 10,000 times. That is not a big deal for me. I will do that if you're not understanding. So let me know. Is it clear? Is it not clear? Cool. If is clear, and if you're here and probably consistently hearing to me, because I do not know who is consistent over here, I cannot see your face, right? If yes, give me a thumbs up and I'll go ahead. And if you're someone who feels that this lecture is actually good, hit the subscribe button. And from the next time onwards, every Thursday I have my class at 4:30 mostly. Please do come for the class and we will finish the entire syllabus together. Cool? Okay, so I think there is time left for you guys to come in numbers. So this is all, and please take a screenshot of this if at all you want it, because this will be lost. Okay? Cool? Yes. Now, we're going to read it once so that everything becomes settled in our head. Spermatogenesis is what? Formation of sperm in the testes starts at puberty. Why at puberty? I'll tell you. There's a reason for that. The hormones become very much active, and the hormone says that, hey, spermatogonia, you have to divide. Okay, this is the basic thing. We're going to learn about the hormones as well. Wait for that. Okay, so spermatogonia, which is diploid in nature. How do you understand diploid? 2n means diploid. Okay, double the number of chromosomes. 46 number of chromosomes are there. Okay, and these spermatogonia which is there, they undergo mitosis. Fine. What do you get? You get the primary spermatocytes, that is 2n. Again, it is diploid only. This primary spermatocyte undergoes meiosis now. It undergoes meiosis 1, you get secondary spermatocyte. The diploid number of chromosome becomes haploid over here. You just have 23 number of chromosomes. Now, then what happened? The secondary spermatocyte again undergoes meiosis 2. Finally, you get spermatids with how many number of chromosome? 23 number of chromosome. Finally, the spermiogenesis process happens where this spermatids converts into spermatozoa. And last process is spermiation, where mature sperms are released from seminiferous tubules. They are released from the seminiferous tubules. That is how it usually works. Cool? So this is the general steps that you need to remember. Do not forget it. And hello Jasmine Mam, how are you? No man, oh my god. You know what, I'll cry right now if at all I see you guys in real. I really miss everyone out there. So the problem is, all of you children usually come towards the examination where you could have actually, like, I literally regret not talking to you guys before itself. So wish we all could have met a little earlier, and this is for 2007 champions also. If you're watching this video, I really wish. I know that in the examination time you will be coming, but let's make it a little early. Come now only. So Norman, I hope you're doing really good. And I'm out of words actually. Cosmic, oh my god, thank you for your guidance, ma'am. I've secured a KCET rank of 2091. Congratulations. Oh my god. Yes. I do remember everyone, obviously. Cannot forget any one of you. So sweet of all of you, and thank you so much, and be at some good place. I'm praying for all of you. You will get the best college out there. Okay? Cool. So now going ahead with this thing also, because I have to complete something. I'm so sorry if I'm just taking it away, but yes, that is there. So this is how it goes. As I said, you have at puberty spermatogonia, it undergoes division, mitosis, blah 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 blah. So I say mitosis happens, the cell basically divides. Basically the cell divides. Okay, now in that one of them, if however now how many number of division happens, it happens, but at least one of them is going to what, develop into what, the primary spermatocyte. So this is the one that is the primary spermatocyte. It undergoes the first meiotic division, you get two cells. What is it? The secondary spermatocyte. The secondary spermatocyte undergoes division, you get round cells. What is it? Spermatids. Spermatids converts into what? Spermatozoa, and that is basically differentiation that happens over here. So here from this point onwards, secondary spermatocyte onwards, it becomes haploid. Please remember that. I am going to give you a question at the end, and those of you who know the answer for this question, okay, you have to, I will be giving you this particular question towards the end, not now. Okay, so you have to tell me once this live ends. You can put it down in the comment section. Cool. Now I have a question for you guys. Those of you who probably know the concept can easily answer this question. Identify the cells representing P, Q, R, S. So this is P, this is Q, this is R, this is S. Okay, in the given schematic representation of spermatogenesis. So this is the spermatogenesis that we just studied, and you have to tell me what is P, what is Q, what is R, what is S. It's easy. It came in 2019. Come on, try it out once. And there is a best way to do that, go backwards, okay? Go backwards. So try to find this and then you will be able to easily answer everything else. So any seniors up there who probably would like to answer a biology question can go ahead and give the biology question as well. Manya Naik says the answer for this question is B. Okay. What is B? B is this. Let's check it out. Is it B? Yes, it's B. And how can you be so correct? So listen to me very carefully. What is B? At the end, we all know the last thing that is going to be formed is spermatozoa, right? And S is spermatozoa. In what all options do you have S as spermatozoa? In this you do not have it. In this you definitely have it. In this you do not have it. In this also you do not have it. The only answer that can be there will be this. Are you the old Mania or this is someone new out there? So in the senior batch also there was Mania, and there could be any Manias out there. So many numbers of Manias. So I'm so sorry. I'm just asking out of curiosity. Okay. So yes, this is how it goes. You have this spermatozoa. That means that who forms the spermatozoa, Baba? It is going to be the spermatid. It is going to be the spermatid. Now, spermatid came from where? Secondary spermatocyte. Q becomes secondary spermatocyte. What about P? P becomes the primary spermatocyte. Got it? So that's why I said go back. New batch, ma'am. Okay, cool. So many manias up there in my life and all of them have been really good. So yes, so good, so good. So Mania gave me the correct answer even after being from a new batch. Awesome, awesome, Mania. You're doing really, really good. So this was a question, and these type of questions usually come. So do understand the importance of the picture-based question. If you do not know the concept here, you will not know the answer as well. Yeah, and you should know the tricks as well. So this is 2019 question. Do we have any more question? I think we have. After every concept, mostly I have given a question. So we will solve that. Ready? Ready for the next concept? And probably we'll go ahead with the next concept then. Cool. So the next concept goes like this. We are going to talk about who gives the signal that, hey, spermatogonia, you have to divide and form the sperms. There should be someone to tell that you have to do that, right? All of a sudden it cannot happen. So basically when I say a guy attains puberty, okay, when a guy hits puberty, there are lots of changes in his body. There will be immense amount of energy as well, and when all of this happens, basically in our brain, a lot of communication happens, and at the end, in our brain, there are two people out there, okay, two people. One person's name is hypothalamus, the other person's name is pituitary gland, okay, and both of them together says that I will manage it, I will manage it, I'll do the whole thing. So we're gonna see who wins and basically what thing is being followed over here. So as I said, we have hypothalamus, cool, and we have one very important thing, and that is pituitary gland. Where is it? Both of them are there in our brain, cool. And in pituitary gland, you need to know there is anterior and posterior lobe of pituitary. So it's not like it's a pair, it's basically two different parts. You call it anterior pituitary gland and you have another part which is known as posterior pituitary gland. So as of now, what I'm talking about is anterior pituitary gland. Okay, yeah. So these two guys are there, and I'm going to talk about two very important hormones also. I have a name for them. I call them gonadotropins. Why do I call it gonadotropins? Basically, they go and act on the gonads. Gonad is what? Where the gametes are going to be formed. So, if, for example, in the case of males, it is testes. In the case of females, it is going to be ovary. So, there are two gonadotropin out there. One is known as LH, luteinizing hormone, and the other one is known as FSH. Other one is known as FSH. These are what? Two gonadotropins which are going to act on our gonads and help in the formation of the gametes out there. Cool. Luteinizing hormone and follicle stimulating hormone. That is a name that we have for both of them. Ma'am, who releases it and why are you talking about it right now? Listen. So basically, hypothalamus says that, hey, pituitary, you have to release the gonadotropins. That means that pituitary guy will listen to it and will be releasing LH and FSH. But before that, When this happens, obviously they cannot talk inside our body. Our brain and different parts of our brain cannot talk to each other. They are going to release certain things and give the information in the form of that. So what does it release? Hypothalamus releases what? They release gonadotropin releasing hormones. This is the name of the hormone. Okay, gonadotropin releasing hormone. You have a shorter name for it. You call it GnRH. That macha, you are having the gonadotropins, you basically release it. So that information is given from the hypothalamus to anterior pituitary with the help of GnRH. This GnRH goes and acts over here, and then the pituitary gland is going to release this, and it is going to release this. Now what do they do, ma'am, over here? Why are they important? Why are you even talking about that? Because children, questions come from this topic and you should be knowing it. And it's considered to be one of the most complicated topics, but I'll try to make it easy for you. Okay. So as of now, we're talking about these guys are going to affect the gonad. That means we're talking about the male part. In the male part, what is the gonad? It is the testis. In the testis, we all clearly know that there are testicular lobules, right? And inside the testicular lobules, you have 2 to 3 seminiferous tubules. Suppose this is one, this can be the next one. These are what? The seminiferous tubule where the sperms are going to be formed. Right, so here the sperms are formed. That means here we have the spermatogonia. Plus what do we have? We also have one more very important thing, very important thing, and that is what? Sertoli cells. Sertoli cells, okay? And in between the space that you see, what do you have? You have another cells out there. What do you call them? You call them as Leydig cells. You also call them as interstitial cells. Why? Because they are in this particular space. You can call them Leydig cells or interstitial cells. Either way, this is all happening in one testicular lobule of the testis. Okay, similarly you have so many. What do you call them? Leydig cells. Okay, ma'am, I got to understand that this is the case. Now what? So, this LH is going to act on the Leydig cell. And it is very easy to remember. Why? Because L over here, L over here. What does it say while it acts? It says that, Macha, you have to release the male sex hormones. That is basically the androgens. You know, the androgen, male sex hormones are known as androgens. The major androgen that we have is testosterone. The major male sex hormone that we have is what? Testosterone. Cool. Why do we need it? Baba, I told you about the process of spermatogenesis, no macha, where this basically primary spermatocyte divide and form the secondary spermatocyte and the whole division happens. For that to happen, you need to give information, or basically you need the help of these hormones. So these hormones which are being released by the Leydig cells, okay, hormones are going to get inside the seminiferous tubules to what? To help in the process of spermatogenesis. So just imagine that right now they are inside, where? Inside the seminiferous tubules where the division is happening and where these sperms are being formed. And for this process you need testosterone out there. Okay, ma'am, got it. What about FSH then? What does it do? FSH has a totally different role. And what is that? FSH says that I am going to act on the Sertoli cells. What Sertoli cells are? Sertoli cells were also there in the seminiferous tubules, right? They are also there. Why are they there? They are the nourishing ones. They are the cells which are going to nourish the sperms out there. That is why they are there. But as of now, why do you need to act on Sertoli cells? What are they going to do with the help of this? So when this communication is done to Sertoli cells are going to release some very important factors. Okay, actually the name of the factor is not given in your NCERT, but I can tell you. Androgen binding factor. That means that these androgens which came in, the testosterone which came in, the Sertoli cells wants to make sure that the level of testosterone is maintained really well, such that the whole process happens really well. You get it? So that is why it is known as androgen binding proteins. So FSH tells Sertoli cells that Macha, you release some factors such that the testosterone is really over here. We saw buttering is happening. So here also buttering is happening. Macha, you be there, because when you are there, you are going to release some factors, and these factors are going to make the testosterone stay such that the whole process is going to happen. That is the basic logic. So I'll repeat it once more. Listen to me very carefully. You have the hypothalamus. Okay, I am the hypothalamus. I came in and like, hey, macha, pituitary, you have to release two things. Okay, you have to release the gonadotropins. What are the gonadotropins? It is LHS and FSH. Okay, I listen to you, but only if at all you release what? Gonadotropin releasing hormones, GnRH. So hypothalamus releases gonadotropin releasing hormones. They go and act on the anterior pituitary gland. The anterior pituitary gland releases LH and FSH. Now LH, which is there, it is going to act on the Leydig cells. Where are the Leydig cells? In between the seminiferous tubule, the space that you see over here. Seminiferous tubule is the place where the sperms are being formed, right? Below that, the space that you have over here is filled with so many cells that is known as Leydig cells. What do they do? The LH, when they act on Leydig cells, it says to produce androgens or the male sex hormones. Wow, the male sex hormone, that is testosterone, is not being made, but does not know what to do. But you know what it is. Basically, it is going to enter into the seminiferous tubule and help in the whole process of spermatogenesis to happen. But if at all you want to maintain their concentration really well, if you want them to stay there and to help in the whole process, you need someone to be there, their best friends to be there. So who is that? Let's decode that. So over here, now the FSH, which was also released, it goes and acts on the Sertoli cells. Okay, the nourishing cells which are there. It acts on the Sertoli cells. Sertoli cells are now going to release some factors. These are the factors that are going to trap the testosterone. They are known as androgen binding factors. Testosterone is also an androgen, the male sex hormone. So you say that, Macha, you stay here, I am also here, you stay here, it's going to be really good. And then the whole process happens very smoothly. But in your NCERT, one line is mentioned, and in that one line, from that one line, so many questions come. That one line says that FSH is super important for the process of, ayyo, don't see, okay, for the process of what to happen, ayyo, for the process of spermatogenesis to happen. What was spermatogenesis, if at all you try to recall? It was the formation of spermatozoa from spermatids. Spermatozoa to spermatids. So I told you, you had your Sertoli cells, right? You had your Sertoli cells and your spermatids and basically spermatozoa. Everything is basically attached to the Sertoli cells, which is providing nourishment. So here, when FSH cells, the Sertoli cells, okay, when FSH goes and acts on Sertoli cells, this Sertoli cells provides nourishment such that basically it helps in the entire process of the formation of spermatogenesis, that is the conversion of spermatids to spermatozoa. So if FSH is not there, who will tell the Sertoli cells to provide nourishment and do all this thing? No one, which is why this process is going to be affected. Majorly this process. Obviously, if FSH is not there, the entire process will be affected, but majorly, okay, but majorly it is this process that is going to be affected. Why? Because of the same reason, because this tells this guy to release some nourishing factors such that it can be transformed really, really well. Got it? This is how it usually works. Have you understood it? All of you who probably are watching this video, tell me. Doubt, Yellamgiri, yes dear, Yellamgiri usually comes in all the classes. Doubt Akka, tell me Yellamgiri. Children, if you are understanding things, let me know. If you're not, let me know. I'm waiting for your doubt. If everything is clear until this particular point, can you give me thumbs up? I know it's a huge lot to take in. That's why if you want to take, if you're someone who is probably watching it later on. Yeah, Varshini. Yes, I remember. Okay, if at all you're watching it later on, you can just pause the video. Okay, you can just pause the video and then go ahead. Subhash. Oh my God, Richard Anthony, Mallu guy. Okay, so cute. Yes, Macha. Cool. So let's go ahead and we miss you so much. Really? I miss you too. I miss all of you. Okay, now there's a question, since I think all of you have understood it, and it goes like this. Where is the question? The question itself went up. Okay. The question goes like this. The process of conversion of non-motile spermatids into motile spermatozoa is called what? Spermiogenesis, oogenesis, sporogenesis, or basically spermatogenesis. Everyone, seniors, juniors, come on, answer this question. This came in 2018. This came in 2018. Richard Anthony, how are you? As far as I recall, you were a senior. D. Okay, D is the answer. Varshini says D is the answer. Are you sure about that? Do you want to give one more thought to it? I'm not asking about the process of formation of sperms. I'm asking what is the process known as with the conversion of non-motile spermatids. That means golu spermatids which were there. Okay, the kids which were there, they're converting into motile, elongated spermatozoa. What do you call that process? I told you, you will get questions on the basis of it. Think. 4 options. One more chance. Come on, Varshini. Come on, other people out there. Mania is still there. If yes, you can also try. Try, macha. No one to try. Which teacher skipped this chapter, da? I don't think anyone skips this chapter. In 10th, in 10th, no, I don't know. Manya says it is C as the answer. Okay, and Varshini says A is the answer. The correct answer for this question is going to be A. Spermiogenesis, I told you. It was globular in shape, papagayi, spermatids. It was known as spermatid, and I remember it as stid as kid. Okay, it was globular. Now it wanted to become the actual elongated sperm. That is known as what? Spermatozoa. And this process, it always wanted to become a sperm. Spermiogenesis. I want to become me. So you can remember spermiogenesis. That is the step that you have, and this is the answer that you have. A is the answer. Even if you have got it wrong, I am pretty sure that you have understood the concept now. Is it clear? Yellamgiri, every class you ask my name. That's not fair. You're forgetting my name, Okay. My name is Jasmine. Yes. And now I am going ahead with the next one. Next topic that you have, I hope this process of spermatogenesis is super clear to all of you. And if you are someone who probably is just watching it in recorded format, I request you to make sure that you come for the live. I'd love it. Okay, and the structure of the sperm is the next topic. Let's go ahead. Let's not waste any time since I did not see any confusions out there. It's a very easy topic. Okay, the structure of the sperm is a very easy topic. What happens over here? Sperm basically has 4 parts. Okay, one is the head, one is the neck. Okay, one is the main body part which is there. I'll say middle piece, and lastly it has a tail. Cool. So basically the head can be divided into 2 parts. This big thing that you see over here is actually the nucleus. Okay, it is going to be nucleus. Yeah, it is the nucleus. At the top of it, over here, you have one very, very important thing. Super important thing, and it is known as acrosome. Why it is super important, ma'am? Basically, this contains enzymes. I'll say hydrolytic enzymes. What is the use of this particular enzyme, ma'am? I don't understand. So the sperm, when it comes in contact with the egg, the egg will be surrounded by a lot of layers, and to go and cleave through these layers, obviously you need some enzymes which is going to open up all the layers, and this finally the sperm nucleus and the egg nucleus can fuse. That is how it usually goes. Okay, so the acrosome contains the enzyme. It's just like the rocket that is there. Okay, it contains the enzyme. Over here you have the nucleus. What type of nucleus? This is important. Haploid. It is not diploid. It has half the number of chromosome, 23. Another 23 comes from where? The egg. There also 23. This also 23. 23 becomes what? 46. Yes, I forgot for a second. Oh my god. 46 number of chromosome. Cool. So that is up there. Now what do you have? You have a neck piece, and you have a neck, and then you have a middle piece. This middle piece is made up of so many mitochondria. And you have always learned, I think this was a meme also that I saw, that no one in their life will forget that mitochondria is the powerhouse of the cell, right? So here also please remember, it's made up of so many mitochondria and it is the power of the cell. Why do you need this much power? Because you need to make sure that the tail which is there is motile, it moves really well, okay? It has to move really well. To provide energy for that, you need the mitochondria lined up over here. So energy source for swimming. So how many parts do we have? We have the head, we have the neck, we have the middle piece, we have the tail. Which part is important for motility of the sperm? The motility of the sperm is basically done by the tail, but who provides the energy for it? It is the middle piece because it has mitochondria. Which part of the sperm has the hydrolytic enzymes to cleave down an egg and probably get into it? It is going to be the acrosome. Which part of it contains genetic material? It is going to be the head, where you have the haploid nucleus. Okay, now always remember, the sperm which is there, it is entirely covered by something. What do you call this something? As the plasma membrane. You call this as the plasma membrane. The sperm which is there is covered by the plasma membrane. Got it? Yes? Done? Yes. Now Varshini says tomorrow class timing. Tomorrow my class is not there, but I think it's math class or chem class. Okay, it will be there at 4:30. Yeah. Now one more thing, Karthik, you don't know, ma'am, in many schools in 10th, even today they skipped the chapter. I don't know. That's so sad if that happens, okay? So wherever I was there, I tried my best to make sure that everyone knew, because it's a topic that everyone should know. I mean, you are kids, but that does not make does not make it any different that you should not be knowing about certain things, which is important, right? So everything that is important, you should be aware of that. We live in an era like that, and no one should be probably facing that discrimination that you should not be knowing about, or probably I should be shy teaching it. That does not work like that. We all are biology champions, right? And basically we should be aware about the body, our body, someone else's body. If you're a male, then you should be aware about how it happens or how it works in the female body as well. Otherwise, you will never understand each other. And basically, this human race is for nothing then. You get it? So, yes. Cool? Cool. Now, very important things are there right over here, and I would love to mention about that. Let's go. Head, neck, middle piece and tail, these are structures. We know that head contains the haploid nucleus and also acrosome. Yes, so do remember, in the head region only you have the acrosome and the nucleus. It's not like two separate things are there. I mean, two separate regions are there. It's in the head only. Cool. Now the middle piece contains mitochondria for energy for the movement. Tail is there for enabling what? The sperm motility, which is very important for the process of fertilization. And now this part is important in terms of question. In one ejaculation, okay, once a male ejaculate, how many number of sperms are basically moved out? It's 200 to 300 million sperms, and you need to remember the number. 200 to 300 million sperms, not billion, okay? It is going to be million sperms that are going to be released per ejaculation. 200 to 300 million. Please not forget it. How do I make you remember this? 3 million. So probably in the next class, if at all you are there, this would be my go-to question, the first go-to question. And if you are someone who probably will be there in the next class, you will be able to answer this as well. Cool? I hope that works. Yes. Now going ahead, 60% of them. So when I say that this much number of sperms are being released, 200 to 300, you should also understand that here in this particular case, 60% should be of the normal shape and size. Right? Normally they should look very normal with the shape and the size which are there, and 40% should be motile for fertility, at least 40%. So 60% of them should be having normal shape and size, and out of that 60 also, at least 40% should be motile for fertility. Because you know what happens? This is very, very important. That is, when the sperm tries to enter the female body, it is very acidic. And female body, it's like anything that tries to enter our body will defend it, would not want anyone to come inside. So what is going to happen? Basically, it will try to kill it. So many sperms die in the entire process. So even though 200 to 300 million sperms are being ejaculated, but only few can get entry. And after that also, there are a lot of processes in the way that only few can go ahead and meet with the egg which is there. So please do know that, for making sure that we do not know who is that few or which one is the few that is going to enter, right? So in this particular case, you should know 60%, at least that number should be maintained. 60% of them should be having normal shape and size, and out of that 40% should be motile. Question comes on this, they can ask you 60% should be motile and 40% should be having normal shape and size. No, ulta, 60% with normal shape and size, and out of that 40% with normal motility. Obviously if you have a normal shape and size, it could mostly be motile also. So you remember in that sense. Cool? Works? Ma'am, what is mitochondria used for? Mitochondria provides the energy for the movement of this particular tail, dear. For the movement of the tail of the sperm. If the movement is not there. So you know what? I had worked in an internship program after my, in the last semester of my MSc, I worked in an IVF clinic. And it was quite shocking to me that it's mostly the men who are having issues with fertility. So in that, ejaculation might happen, but the sperms which are being formed or basically produced, they are not motile at all. In that particular case, do you think it can travel and go meet with the egg out there? No. So this is one very important thing, and this could happen. Why? Because mitochondria and everything is not there properly. Cool? Ma'am, is that spermatozoa in book where there is four tail when we draw diagram used? Four tail? What is that? I do not know, Karthik. Sorry. Okay, now there's a question over here and it goes like this. So in the given diagram of human sperm, identify the functions of the labeled part A, B and C. So A is given at the top, okay, B is right over here and C is over here. You have got the options, you have to choose which one does what. Yes, Varshini, tell me, Akka. Which one does what? A. A is the answer for this question. Let me see if Varshini is correct or not. Others, please do try. Please do try. Let me see if A is the answer or not. A means, A is what? So first thing that we have to do when we get a picture based question is tag them. What do they mean? So this is acrosome. Okay, this is going to be the middle piece which is there. This is going to be the tail. Now I know. A, acrosome, helps in penetration of sperm into the ovum. Correct. B, that is middle piece, helps in the movement of the sperm. Not sure, that could be tail. C, provides energy. So this is wrong. Why? Because it is said that middle piece helps in the movement of the sperm. No, middle piece provides the energy. You have got the next option, you have to see that, right? Right? So this will be wrong option. Okay, going ahead with the next thing. Helps in the penetration of sperm into the ovum. Okay, fine. A is correct. B is middle piece, provides the energy for this movement of the sperm. C is what? Helps in the movement of the sperm. Tail helps in the movement of the sperm. This provides the energy, which is why your answer would be B. But still we have to look forward for the next options. A, helps in movement of the sperm. Cannot be true. It's not for movement of the sperm. A, provides energy for the movement of the sperm. Does it provide energy? No, Baba, that is mitochondria. So again, that would be wrong. Are you getting it? The only answer for this question that you have is this. And as I always say, whenever you solve question, no Baba, no Varshini, the answer for this question is going to be B. Do you have doubt? Do you have doubt? Please tell me if you have doubt. I am ready to take that up. No problem. This is a very important concept and you should not be getting it wrong. I have no idea, dear, why four tails. Okay, so yes, Varshini, tell me, doubt. What is the doubt? Ask me, is this the doubt? This question the doubt? Here you have A, no. A is what? Acrosome. And we know acrosome is a structure which has the enzymes to penetrate in the ovum. Right? So I have already mentioned it that A is what? Acrosome. Cool. B is what? It is the middle piece. What does the middle piece have? It has lots of mitochondria. Middle piece, M for middle piece, M for mitochondria. Then what is C? C is going to be the tail. Listen, tail is going to help with the movement. It is the one that moves. Who provides energy for it? It is going to be the middle piece because it has the mitochondria. Now over here, when I see these many things and I see the option, A helps in penetration of the sperm into the ovum, that is correct. What about B? B is middle piece. It helps in the movement of sperm. Still, I'm not sure whether this answer is correct or not. But when I read the next one, C provides energy for the sperm. Now, tail cannot provide energy for the sperm. C cannot do that. It has to be B only that does that. Which is why option A would be wrong, because here it is tagged in a wrong manner. Now, what about B? A helps in the penetration of the sperm. Correct. Acrosome does. Right? The next one is what? Provides energy for the movement of the sperm. Does B provide energy for the movement of the sperm? Obviously yes, it has the mitochondria, it will provide the energy. That is correct. And lastly, C is the one that helps in the movement of the sperm. That would also be correct. Got it? Now did you understand it? Is it okay? Yes, perfect. Thank you so much. Cool. Now we are going to get ahead with the next topic, that is oogenesis. And I had already told you one thing, that is, in the case of male reproductive system, the first class that we had, the sperms which are coming from the testes out there, they are going to received by so many glands. Seminal vesicle will be there saying that, hey, take fructose. Prostate gland will be there, says that, hey, take calcium and enzymes. Bulbourethral gland will be there, says that, hey, I will be helping in lubrication. So basically, all these three glands, we have three accessory glands which are going to receive the sperm and provide it with some or the other thing. We say the secretion of all of it together, it is known as seminal plasma. Okay? Plus the sperm, when they come together, I will call this as what? The semen. I will call this as semen. So please do remember that there's one extra thing that I wanted to tell. I have told it before also, but yes. Cool? Yes, works. And now we are going to go ahead with the next topic, and that is oogenesis. Watch is beautiful, Akka. Thank you so much, dear. Okay. Can we go ahead with the next topic? Is everyone okay? Okay, oogenesis. After that, it's going to be the end of the class. You do not have to be worried. I started the class a bit late today, I think 4:45. I would have to stretch it a bit late, little late. By 6 o'clock, obviously, I'll end it up. Okay, so please bear with me. Let's just complete this topic and then go ahead. Yeah, if yes, can you give me a thumbs up? If all is cool and we are going to go ahead with oogenesis with a good mind, can you give me a thumbs up? Quick thumbs up. No thumbs up? No, ma'am, we are not gonna listen to you. Oh, okay. I got a thumbs up. Thank you so much, Manya. Okay, cool. Let's go ahead. So, in oogenesis, things are going to be different. Little different, okay? So, in spermatogenesis, we saw that a man, when this, Yarshini also, thank you so much. Okay, when this particular guy was, basically, when he hit puberty, that's when the whole process started up. But in the case of females, things are going to be different. The moment it's a female which is there in the mother's womb. Okay, suppose I was in my mother's womb. At that particular time itself, in the ovary, you would be having oogonia being formed. The oogonia already there. Oogonia is what? The female germ cell. The female germ cell. This is the one that wants to form the ovum later on. So you can just remember like that. Okay, so we have oogonia being formed when we are in the fetal stage. Okay, when we are in the fetal stage, at that point itself, almost millions of oogonia, not one, there will be what? Millions of oogonia which is going to be formed out there. And here also the same thing happens. The oogonia is going to divide, mitosis, it is going to increase its number. Okay, and there also spermatogonia divided, increase the number. And I told you, one of them, okay, if I say one, there will be so many obviously, where the oogonia which is there, it divides, and later differentiates into something that is known as what? The primary oocyte. It is known as the primary oocyte. It is the one that is going to form the ovum later on. So I'll just repeat it because I think it's confusing when I heard about it. Listen. So you had oogonia, how many? Millions of oogonia. These oogonia are dividing with the help of mitosis. Okay, just imagine there is one. Okay, there is one. There are so many which are doing it. I'm just focusing on one. This one differentiated to form what? The primary oocyte. Differentiated to form what? The primary oocyte. What is the number of chromosome over here? 2n. Okay, the primary oocyte said that, Macha, I want to become an ovum. And we all know how things work. The point from here, when something becomes primary, there also primary spermatocyte, when it became primary spermatocyte, the next thing is that it had to undergo a division. What was the division? It was known as meiosis. It was known as meiosis. So same thing happens over here. It is going to undergo meiosis, and meiosis happens in 2 stages. So over here, okay, over here, you will get 2 cells at the end of meiosis. Just like that, in spermatogenesis also you saw primary spermatocyte divided into secondary spermatocyte. Here also that happens. You get secondary oocyte. But there is a twist. And the twist is that after the end of meiosis, yes, it was diploid here. 2n number of chromosomes was there. It becomes haploid over here. That is one thing that we already know. But one more thing happens, that is, this division is going to be an unequal division. So one cell is going to be a normal cell. The other one, which is there is a small cell out there. You call it the first polar body. You call it what? The first polar body. What do you call this as? You call this as the secondary oocyte. After the end of meiosis 1. There both of them were equal. Here that is not the case. That is the difference. First difference what? There it happened at puberty. Here it is happening in the fetal stage only, when the babies are there in the mother's womb itself. Okay. The primary oocyte, when it undergoes division, when I say meiosis 1, at the end of it you get secondary oocyte and you get unequal cells. One small cell, one big cell. This small cell is known as first polar body, and here you have the secondary oocyte. Now what happens? You need one more meiosis to happen, right? So let's do that. One more meiosis happens over here. Okay, again the same thing. One small cell and one big cell. Again, it is going to be what? As I always tell you, the first round of meiosis is where it becomes half. After that, it remains the same. The number of chromosomes are going to remain the same here also. It was n, here also it becomes n. What do you call this? This was the first polar body. This is known as the second polar body. This is known as the second polar body. And what do you call this as? This is our main guy, and that is known as what? Ovum. It is known as ovum. So you should understand one difference. There at the end of spermatogenesis, we got 4 sperms. We got 4 sperms. If at all you remember, it looked like this, right? One cell over here. One primary spermatocyte over here divided, and at the end of it you got 4 sperms. But here that is not the case. Here when I say that one primary oocyte is dividing, at the end of it you get only one ovum. Always remember, this can be a question. If at all they are giving you 200 oogonia there, how many ovum will be formed? 200 only. It will be the same number. But if at all they are asking you 200 spermatogonia is there, how many sperms is going to be formed? So 200 into 4, 800 sperms are going to be formed. So just know that. I'll make you solve a question with which you will remember things a lot better. Okay, this is how the usual division takes place. But you should know that in the case of females, it's not like tuck, 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 tuck division happened. It takes years for all of this thing to happen. Okay, and to make you understand certain thing, let me go ahead and explain that in detail. So when I say that meiosis happens over here, and meiosis 1, in each round of meiosis you have prophase, metaphase, anaphase and telophase. And this is prophase 1, metaphase 1, anaphase 1, telophase 1, right? Similarly here you have again prophase 1, metaphase 1, anaphase 1 and prophase 2, metaphase 2, anaphase 2 and telophase 2, right? That is how meiosis usually happens. When I say the primary oocyte is going to undergo meiosis, one very sad thing happens. It gets stuck. It gets stuck at this particular stage. It is not able to divide any further to form this or this. It gets stuck in the prophase 1 stage. Prophase 1 stage. And when does this happen? when we are basically again in the fetal stage itself. Right now we are inside our mother's womb, females, okay, we are inside the mother's womb. Oogonia is there. Oogonia divided, increase their number. There are millions of oogonia, and then basically they are converting into what? Primary oocyte, or they're differentiating, okay, they're maturing, not converting, I'll say differentiating into the primary oocyte which is there. Now the primary oocyte thinks that I can divide and form the ovum, but what happens once it starts undergoing division, it gets stuck in a phase, and that is known as prophase 1 phase. Okay, and it remains in that particular stage until we hit puberty. So all these things are there in our ovary. Okay, the primary oocyte is there in our ovary like that only until we hit puberty, and there will be a lot of them which are going to die off, which will be dying off. So we started off with millions of oogonia, probably millions of primary oocyte as well, but later towards the end, it's not going to be there. Only few of them are going to be there in each of the ovary. Okay, and what is happening, as I said, it is stuck in this particular stage. So few more things happen in the background, and I'm going to talk about that. So make sure you listen to me. Okay, you listen to me. So over here, do I have a page, extra page for explaining you this? No, I don't have a page. That means that I have to explain this. Cool, let's explain. So we had the oogonia, and then we have got the primary oocyte. The primary oocyte started off the division but is stuck in which phase? The prophase 1 phase. Suppose this is the primary oocyte that we are having, and it is diploid in nature. So what happens? It has to sustain until, okay, it has to sustain like this only until we hit puberty. This is from birth until the time we hit puberty. It is stuck in this particular phase. But you need to understand one very important thing, that basically to make sure that this particular cell is being safe, you have layers of granulosa cell being formed over here. You have a layer of granulosa cells being formed around over this, and you call the whole thing as what? The primary follicle. What do you call this as? You will call this as the primary follicle. So do remember, primary oocyte which is there, it's not alone. It says like, Macha, don't feel sad. I am here to protect you. I am the granulosa cells. And basically, you and me together, we will call us as what? The primary follicles. So we say from birth to puberty, there are so many of primary follicles which are going to degenerate, which means that the primary oocyte is also not going to be there. By the end of it, we only have a few of it, that is 60,000 to 80,000 primary follicles in each of our ovary, not in one ovary. In each of our ovary, we will be having 60 to 80,000 primary follicles out there. Okay, now once we have hit puberty, what is going to happen? There will be, please repeat again. Okay, I'll repeat again. Listen to me very carefully. You had the oogonia divided to form the primary oocyte, right? Primary oocyte wanted to form the ovum by division, but then a twist happened. While it underwent, it started to undergo meiosis. Someone said, and like, Macha, you cannot go ahead. First of all, you have to hit puberty. You be there in prophase 1 stage only. When you hit puberty, we'll think of continuing the meiosis, and basically then you form the secondary oocyte and things, whatever you want to make. So you said like, okay, fine, then there should be someone to protect me. And that's exactly why you have granulosa cells which nourishes it, make sure it's safe. And this particular structure, that is granulosa cells plus the primary oocyte, you call this as what? The primary follicle. So many primary follicles are there from birth to until the time we hit puberty, but that time, lots of them, they get degenerated also. You almost have 60 to 80,000 primary follicles in each of your ovary. Done. Now we have hit puberty. Okay, obviously under the influence of hormones, certain things are going to change. And what is the change? What is the first change that you are going to observe? This is, and so basically hormones are going to come in play when we hit puberty, right? So you had the primary oocyte remains the same only. No change. Okay, it remains the same. But the granulosa cells which were there, this was only one layer, right? Now it becomes two layer, multiple layer. It becomes one more layer, and apart from that, you have one extra thing coming up there which you call as what? theca layer. So there is theca layer and there are multiple layers of what? Multiple granulosa cells layer. The internal thing, is it remaining the same? Is it remaining the same? It is going to remain the same. Basically, it is the primary oocyte itself, just that it has a separate coating, and now we will not call this as a primary follicle. You are now going to call this as what? The secondary follicle. You are going to call this as what? The secondary follicle. Why did we have millions? So it's just like you have seen dogs having so many puppies. Why they have so many puppies? Because they are pretty sure that a lot of them are going to die. Similarly, it was millions in number. We are pretty sure that from birth to puberty, a lot of them are going to die. And these follicles, the granulosa cells which are there, they are there to nourish all the papa guy, that is the primary oocyte which is there. Okay, just to provide nourishment, but still a lot of them die. Okay, the ones that survive, few of them under the influence of the hormones out there, they are going to, now there are so many primary oocytes, just you have to remember that, and out of that, under the influence of the hormones, what is going to happen? Some of the primary follicles are going to develop into secondary follicles. When I say secondary follicle, what is the change that you have? You have an extra theca layer being formed, and also you have multiple granulosa cell layers also being formed. Cool, cool. Now again, these hormones are at play. Right, these hormones are still at play. So what is going to happen? These cells, okay, this particular follicle that you see, the secondary follicle that you see, is going to change. Is going to change. What happens to secondary follicle? Let's see. So secondary follicle is there, and you have the primary oocyte right over here, right? So I'm just going to erase it a little bit, so that I find a little space. So I'll call this as a secondary follicle. So this was the first one, this is the second one, and now I'm making the third one. So over here, the granulosa cells are still there. Okay, got it. But along with granulosa cells, you're going to see something new. There is something like this which you call as fluid-filled antrum. So every time some change is going to happen, it is what it is known as the fluid-filled antrum or the antral cavity. And you remember the theca layer? The theca layer differentiates into two things: inner theca interna and outer theca externa. Inner theca interna and outer theca externa. Two theca layers are being formed, and what do you call this structure? After secondary follicle, what do you can expect? What can be there? Anyone who is watching this video, what can you expect? After the secondary follicle, first we had primary, then we had secondary, and now we have what type of follicle? It is going to be tertiary. It is going to be what? Tertiary follicle at the end. Tertiary follicle at the end. Now why this is happening? Because we are under the influence of hormones. Is all the primary follicles converting like this? No. Only some of them are going to. The ones which are remaining, 60 to 80 are remaining now. Out of that, only few of them during each cycle of our menstruation, my dear children, under the influence of hormones, they are going to convert into secondary follicle, and the secondary follicle is going to convert into what? Tertiary follicle. Why do you think this conversion is happening? Because now we want one thing, very important thing to happen, and that is we stopped the primary oocyte over here at this stage, prophase 1. We have to make sure the primary oocyte undergoes division. Is it visible? Yeah. Undergoes division and forms the secondary oocyte and the first polar body. And this is exactly where it is going to happen. Here inside the tertiary follicle, this very big change happen, that is, for the first time, it is going to divide into what? The secondary oocyte, and there will be the first polar body being formed. This happens where? It is going to happen within the tertiary follicle. Right? Now the tertiary follicle is out there, it's all cool. We have formed the secondary oocyte, but still we are left with one thing, and that is what? This secondary oocyte which is there, again it has to divide and has to give me the ovum at the end, N, and the second polar body. When do you think that happens? Let's check it out. So the tertiary follicle, again under the influence of the hormones, it is going to divide into something else. But first of all, you need to know that it has formed the secondary oocyte, and the secondary oocyte now thinks that, okay, until now the follicles were doing everything for me, but now I have been formed, so I have to do something for myself to make sure that I am protected. Okay, and what is that thing that they do? Always remember, this is going to be super important. So basically, suppose this is the secondary oocyte that we are having. It is haploid in nature. What happens to the first polar body? It usually gets degenerated. Why do you think that first polar body is very small? Because whatever cytoplasm was there, it was taken by this guy. It is going to form the ovum. A gamete needs to be super good, right? It took all the cytoplasm for nourishment. Okay, so now we had the secondary oocyte. Secondary oocyte says that, Macha, I'm going to form a new layer around me. And the name of this layer is super important. What is the name of this layer? It is known as zona pellucida. It is known as what? Zona pellucida. It is a non-cellular layer. That means acellular. It does not have any cells being formed. It is what? Acellular layer. And it is made up of what? Glycoproteins. That means basically carbs and protein. Glycoprotein, that is what it is made up of. Yeah, and surrounding it you will be having what? You would be having some granulosa cells. You will be having some granulosa cells. So what do you have inside? First is zona pellucida. Then around it you have corona radiata. These cells, basically these cells are granulosa cells. These are not being formed by the secondary oocyte. Secondary oocyte forms just one layer, and that is zona pellucida. Okay, the corona radiata that I am talking about basically comes from the granulosa cells itself. So this is known as corona radiata. Okay. Apart from it, what do you have? You have everything else which was already there. You had over here the granulosa cells, right? This is still there. The antral cavity which was there, it becomes very big. It becomes very big. And finally, you have the theca layers also. You have the theca layer interna, theca layer externa. What is the major thing that you need to remember? First of all, what do you call this as? You call this as the last one, and that is known as the Graafian follicle. It is known as the Graafian follicle. Cool. And the Graafian follicle, the major change that you see is the secondary oocyte which is being formed. It forms a layer around it. This is known as zona pellucida. Remember, it's a acellular layer. No cells are there over here, and is made up of what? The glycoprotein. It is made up of glycoprotein out there. These two things are very important. Along with that, they also have corona radiata around it. That is, the cells are being around it. These corona radiata is not being basically formed by the secondary oocyte. Only one layer is formed, and that is zona pellucida. These cells which are there basically comes from the the granulosa cells itself. Okay. Apart from that, this particular tertiary follicle which was there, it becomes a little more bigger. So this is known as the Graafian follicle. I will also call this as the mature follicle. It is also known as the mature follicle. So when I say that a female is ovulating, okay, when a female is ovulating each month, half of the month she is going to, by the half of the month she is going to ovulate. When I say that she is going to release the egg, you need to understand one very important thing. Has she completed the division? Has she actually formed the ovum over here? This to this division happened where? This to this happened in the tertiary follicle. You have finally got the secondary oocyte over here. But the secondary oocyte has not completed the division to form ovum. So when I say that ovulation happens, you need to understand that the Graafian follicle which is there, this big follicle which is there, it is going to rupture. And the secondary oocyte is the one that is going to be released, not the ovum. Why? Because the secondary oocyte is also stuck in the stage. In which stage? Metaphase 2. So here this was stuck in which stage? We said prophase 1. So we have prophase, metaphase, anaphase, telophase, 1, 1, 1, 1. It was stuck here. Then it underwent division and finally formed the secondary oocyte where? Here when it formed the tertiary follicle. Right now, sorry, tertiary follicle. Right now when I say secondary oocyte, again when it goes divides, Baba, it is going to be stuck again. Okay, now this time it is going to be prophase 2, metaphase 2, anaphase 2, telophase 2. Right now it is stuck over here in metaphase 2. Okay, it has not formed the ovum. When do you think the ovum is going to be formed? When the sperm, so this egg is being released and the egg waits over there. Basically, it's not the egg, it's the secondary oocyte. We generally said ovum is released, ovum is released. No, in ovulation, the Graafian follicle ruptures and secondary oocyte is being released. Okay, now the secondary oocyte is waiting for the sperm. When the sperm comes, there are going to be some changes around it such that finally it can undergo division and form the ovum. It can undergo division and finally form the ovum and the sperm are going to fuse. Okay, so first it comes in contact with the secondary oocyte. There are going to some changes out over here. It forms the ovum, it undergoes the division, it forms the ovum and finally fuses with the nucleus of the ovum. That is how it usually takes place. Got it? I'll just rewind whatever I started over here and probably that is going to give you some clarity if you're someone who is lost somewhere. Okay, first thing, we're talking about the female body. In the female body, while suppose I am the baby out there and I am inside my mother's womb, within my fetal ovary itself, millions of oogonia are going to be formed. These oogonia which are being formed, they want to form the ovum later on. At the end, they want to form the ovum. So they say like, let's differentiate into primary oocyte. That means primary oocyte is a guy that can later on form the ovum. So let's differentiate and let's mature into that. Okay, fine. I have become the primary oocyte. How many of them? Millions of them have become the primary oocyte. Okay, primary oocyte tries to undergo meiosis, but what happens is that there's a tragedy that happens. It gets stuck in what? The prophase 1. You have prophase, metaphase, anaphase, telophase. It gets stuck in prophase 1. So the primary oocyte is just like that. And to nourish it, you have so many granulosa cells that comes around it. Okay, and when it comes around, you will now call this as the primary follicle. It remains as a primary follicle until when? Until we hit puberty. And by that time, we had millions of them. Some of them degenerated. We only have 60 to 80,000 follicles remaining in each of the ovary. And they are there. Right. Now, the time when we hit puberty, what is going to happen? Some of them, not all of them, some of them are going to be influenced by the hormones, right? The hormones, the FSH and the LH, and a lot of things are going to be coming. Don't be worried about that. Okay, because of the hormones, what is going to happen? There are some changes happening in the primary follicle. The primary follicle converts into secondary follicle. What are the major changes? Basically, you just have one layer of granulosa cells. Now you will be having multiple granulosa cells. And apart from that, you have a new layer being formed that is known as theca layer. This is a feature of secondary follicle that is there. Now you have formed the secondary follicle. Again, you are under the influence of hormones only. So this secondary follicle which is there converts into tertiary follicle. What is the new change that we have? You had only one theca layer, right? This time it's two of them. The inner one is known as inner theca interna and the outer one is known as outer theca externa. Okay, apart from it, what do you have? You have a fluid-filled cavity. What do you call this as? Antrum. You call this as antrum. And one of the major changes that happens is that basically the primary oocyte which was stuck in prophase 1 will be able to undergo this division, will be able to complete anaphase, will be able to complete telophase, and finally forms what? The secondary oocyte right over here. And what? And the first polar body. This is an unequal division that happens. Basically, it forms both of them right over here. This papa guy gets degenerated. This guy took all the cytoplasm out there. Now what happens? Again, under the influence of hormones. Okay, under the influence of hormones. Yes, Hemant, is there something that you want to ask? Okay. Cool. So over here, under the influence of hormones, finally, again, it forms the Graafian or the last follicle, which is known as the mature follicle out there. Right? And the mature follicle, you need to understand, has formed the secondary oocyte, right? Secondary oocyte has been found and says that, Macha, I need my own layer. Okay, you did all of this things for me, but I have to form my own layer for my protection. So basically, what do you have? The secondary oocyte is going to have its own layer, which is known as zona pellucida. It makes it by itself, and it is acellular layer. There are no cells involved. It is formed of glycoproteins. Glyco, basically carbohydrates, and protein, protein, right? So glycoprotein layer is being formed around it. Along with that, you see this yellow thing. Usually, it's not yellow. I'm just making it. Basically, this yellow thing is also being formed. Basically, they are cells being grouped up together. You call it as corona radiata. Corona radiata. Around it, you have everything just like how you had it. Now, why do you think that secondary oocyte made all of it? Okay, why do you think it had zona pellucida being made by itself? Why do you think it allowed the coronal radiata thing to be formed? Because the next big thing that is going to happen is, we are talking about the influence of hormones. We are just studying about oogenesis, but you have to mix it with menstruation. In menstruation cycle, after every half of this, so I say 28-day cycle is there. When we reach the half of the cycle, what is going to happen? The graphene follicle is going to rupture, and the secondary oocyte which is there, it is going to be released. While it gets released, there should be protection, and that is why it has zona pellucida around it and also corona radiata around it. That is how it works. Now the sperm will come. It is going to use its acrosomal enzymes to cleave through the zona pellucida, corona radiata, and go and fuse with the secondary oocyte. And before it happens, the secondary oocyte which was stuck in this phase, that is metaphase 2. So prophase 2, metaphase 2, anaphase 2, sorry, telophase 2, anaphase 2. It was stuck in metaphase 2. It was not able to complete the division. Finally, it is able to complete the division. It forms the ovum as well as the second polar body. And while it does that, just after that, the sperm nucleus and the ovum nucleus is going to fuse, and it forms the zygote. This is what usually happens. I hope now it is better, it is more clearer to you guys. If you have any confusions around it, you can ask me. And one more very important thing, I will not be taking any spammers over here. So yes, I do have the access, okay, to remove anyone who comes in spams for no reason. Listen, I'm not at all a person who can be very strict with you or can be strict at all. I never liked it. But there are some fake profiles that comes in probably and say some rubbish stuff. Obviously, we cannot be promoting them. We have to block them up. So this is the usual practice which is there. And as I said, you will get to know more about it. When it's exam time, there will be so many children watching this video, and some out of them, they are not children, they are someone else. They probably come here and talk anything and go. Basically, we would put them in block mode or probably timeout. So yes, I will be doing that. So do not ever talk rubbish. Okay? You can say you're not understanding. Definitely, I will help you on understanding. But nothing that is going to disturb the morale of the class, that is going to be so sad. I'm going to be sad. You're going to be sad. Why to make that, right? We're a happy class. Let's make it a happy class. So children who are watching this, have you understood the whole thing of oogenesis? Do you have any doubts around it? Again, if you want to take a screenshot of this, you can definitely go ahead and take. Cool? Yes. Now, so I'll just read it out once so that we are not missing out on anything. It begins during embryonic development. A couple of million oogonia, that is gamete mother cell, form in the fetal ovaries. In the fetal ovaries, they are going to be found oogonia. No new oogonia are added after birth. After birth, no, none of them are going to be added up. Okay, it's not like after we are being given birth, there will be few more. No, none. Oogonia enter the prophase 1 of meiosis and get arrested, become primary oocyte. So over here it has been written like that. So in general, we can just remember it will be transformed into primary oocyte and it gets stuck in prophase 1 of meiosis. That is very important. That you need to remember, it gets stuck in prophase 1 of meiosis. This part is very, very important. Meiosis 1, prophase 1. Yes. Now each primary oocyte is surrounded by granulocyte cells, becomes the primary follicle. Correct. Many follicles degenerate from birth to puberty, leaving only 60,000 to 80,000. These things are important. This is important. 60,000 to 80,000 primary follicles is important. Now the primary follicles convert to secondary follicles, more layers of granulocyte cells, and theca formation happens. Theca layer is also being formed, more granulocyte cell layers are also being formed, just like how we discussed it. Now later on what happens, the secondary follicle transforms into tertiary follicle, develops a fluid filled cavity, that is antrum. This is something new. And what happens to the theca layer? It differentiates into theca interna and theca externa. That also happens. Clear? Yes, ma'am. Now the primary oocyte completes meiosis 1 finally and produces what? Haploid secondary oocyte. Right? It was diploid until now. It produced haploid secondary oocyte and the first polar body, which is small, and it can be degenerated later on as well. Cool. Now, Now what happens? You got that graphene follicle development and ovulation. Tertiary follicle which is there converts into graphene follicle, which is the last or the mature form of follicle out there. Secondary oocyte forms zona pellucida layer around it. In the graphene follicle, what is happening? The secondary oocyte is forming a new layer around it, and that is known as zona pellucida. Very important. It is what? A protective glycoprotein layer. It is acellular or non-cellular. Please remember, it does not form of any cells. Okay, and there is no mention about corona radiata. Please know there is corona radiata. That is a layer of granulocyte cells that forms around it. Cool. And then graphene follicle ruptures, ovulation occurs, releasing the secondary oocyte into the fallopian tube. Ta-da! And everything is being done. Cool. Now over here, there is a question that I have for you guys to solve. This came in 2017 KCET examinations. In human females, number of primary follicles left in each ovary at puberty is what? In female, the number of primary follicles left in each ovary at puberty is what? 3,000, 30,000, 30,000 to 60,000, 60 to 80,000, or 1,50,000 to 1,60,000. What do you think is the answer for this question? Any idea? How many number of them of follicles are remaining in the ovary? That is the question. At puberty. C. Mania says the answer for this question is C. Perfect, Mania. So I'm pretty sure that you have got an understanding of it, and I'm super proud of you. Yes, so this is a picture. Again, this picture is also something that you have to remember. So always remember, at this side you have the primary follicle, okay, and if you look at the picture, okay, this is the primary follicle. You have the antrum for the first time, that has to be tertiary follicle. The secondary follicle is mixed over here. Then after the tertiary follicle, you will be just having what? The graphene follicle, you know that. And I told you the graphene follicle is going to rupture to produce what? Not the ovum. In your NCERT, they write it like that, but it's actually secondary side. But it's okay. Okay? It ruptures. Now whatever is left behind, you are going to study that in the next class in menstruation. It is known as corpus luteum. It is known as corpus luteum. So this will be the last part. Why I am telling you about this particular diagram is that you can get questions based on this diagram as well. Yeah? So please do remember that. Cool. And it happens, like so many of the primary follicles are going to be matured, but only one is going to form the graphene follicle later on. Okay, only one forms the graphene follicle later on. Just everyone stops, like Macha, you stop. It will be of waste, but yes, that is how it usually happens. And when all of these primary follicles are done, we say that the whole process is over. Menarche, we have reached menarche. There is no more ovulation that can happen at all. Menstruation is stopped when the follicles are completely done. Now, comparison between spermatogenesis and oogenesis, as I told you, here it happens in the puberty. At the puberty it starts. Here it starts from the fetal stage itself. Here at the end of it, you just get one ovum. But here you are going to get 4 sperms. Very important. Here when you say the primary spermatocyte divides, it divides into 2 equal cells. You call them secondary spermatocytes. They are haploid in nature. But here when that division happens, it is going to be unequal. One is going to be first polar body. Second one is going to be the, secondary oocyte which is there. Same thing happens over here as well. So always remember that the ratio of this, okay, you might be asked the ratio of this, it is what? 4:1. 4 sperms here and only 1 ovum here. If you're given the question, 200 oogonia is there, how many ovum is going to be formed? 200 only. 1 oogonia, we consider it will be forming only 1 ovum. Here in that case, if I say 200 spermatogonia are there, how many sperms are going to be formed? 200 into 4, 800 sperms are going to be formed. This is how the calculation works. And I think that's all. So I have not put that question over here. But this is a homework question that I have for you guys. Make sure you solve it and do let me know. Not now. I don't want the answer right now. You can do let me know in the comment section when this live ends. Okay, and I will be taking the screenshot of those of you who gave me the answer. And before that, one more very important thing, we are launching Pariksha Veera course. We have launched it already, and the classes are going to begin from the month of July itself, this month. Next Monday, that is July 6th, your classes will be starting. Make sure you check it out. This is a course where your complete 12th syllabus or your PU2 is going to be completed in by November 6th. That is the deadline that is there, and the pricing for that is rupees 3,499. If at all you want to know more about it, the link will be given in the description section as well. Cool. So I think that's all about today's session. I hope that it was helpful, and I hope that you had understood the concepts, right? Is there any doubt that you want to ask me, or is it over? Like, okay ma'am, it's done, understood, just that you have to do your revision. Tell me that, and probably I'll also wind that up and I'll also go. Okay, I think it's good time to go. Fine, I do not see any doubts coming because there are no much children watching this video as well, but I am pretty sure it will be coming towards examination. That is how it usually happens. So that's all then. Please have a very good day, and as I said, every Thursday be there for the live, and lots of love. Bye bye. Do not forget to hit the subscribe button as well, and bye Mani if at all you're watching this, and also Varshini. Bye bye. Take care.